Am I audible to you all? Good morning, children. So today what we are going to start with the transforming that is by the formation of nucleosome. The DNA strand takes then there are about 3 into 10 raised to the power 7 nucleosome in a diploid human mammalian cell, human cell. Then we come to the next topic, and it is the search for genetic material. In this, the first topic which we are going to discuss today is transforming. So this is what we are going to discuss today, which is transforming principle. Now here in this principle, this transforming principle, or the principle of transformation, it was given by Frederick Griffith in the year 1928. So in the year 1928, Frederick Griffith, Griffith. He was a researcher who gave this principle, which is worked on the bacteria, and the name of the bacteria is Streptococcus pneumoniae. Worked on the bacteria named Streptococcus. This was the bacteria on which he worked, Streptococcus pneumoniae. He cultured this bacteria. When he cultured this bacteria on a plate, in the lab, he found that there were two strains of this bacteria. One was R strain, which was the rough strain. And another strain of this bacteria was S strain, which was the smooth strain. He noticed that in the R strain, the surface is not shiny and smooth. And this bacteria, because of non-shiny and rough surface, this bacteria has non-shiny and rough surface, and this bacteria was non-virulent. Non-virulent bacteria means the bacteria which does not cause a disease, and this was the rough colony that it produced, and it was non-virulent. And this smooth shiny surface was because of the formation of a mucus layer of polysaccharides. And because of this, it was virulent. Virulent means which cause infection. This is not lethal, this is non-infectious. And this one is infectious. infectious and non-infectious. That means if infected by R strain, the organism will not suffer from the disease and will not suffer from the disease pneumonia and will not die. And if infected by S strain, the organism will suffer from the disease pneumonia and will die. So he found there were two colonies of bacteria, Streptococcus pneumoniae. One was R strain, one was S strain, one was virulent, and one was non-virulent. Virulent means which causes disease, and the one which does not cause disease. That means if an organism is infected by S strain, it will die because of the virulence, because of the disease. But this is in case of animals. In case of human beings, it is not there. If any human being is infected by Streptococcus pneumoniae, the person will not die. But if the person is given treatment, it will remain alive. Like same way, there are many diseases in which the person doesn't die because they are saved by the treatment. Now here we are talking of experiment which was done using the experiment, and that he took this S strain. S strain bacteria was taken in an injection. S strain bacteria was taken in an injection, and then this S strain was injected in mice. Injected where? In mice. So what happened to the mice? The mice died of pneumonia. Because this S strain is virulent strain, or the strain which causes pneumonia. So the mice died of pneumonia. Now, what he did, he took another strain which was R strain. This R strain was also injected in mice, and the mice remained alive. The mice remained alive because R strain was non-virulent or non-infectious strain, does not cause the disease. So the mice remained alive. It did not die. Now, further, what he did in the experiment, that he took the S strain and killed it by heating. S strain, which S strain he took? This S strain, which was virulent strain or infectious strain or which causes disease, he took that strain. And this S strain, what he did, he killed it by heating to that limit that the S strain bacteria died. So then, when the S strain bacteria died, he took heat-killed S strain. S strain bacteria were taken, but these bacteria were not alive, these were heat-killed. These bacteria were heat-killed. So when he took this heat-killed S bacteria and injected it into mice, and when these were injected into mice, there were the infectious causing agent died because of killing by heat, because this S strain was heat killed. When it was injected into the mice did not die, it remained alive. It did not die because it did not cause the disease as it was heat killed. So it did not die. Then what he did in his another experiment, he did one more thing, that in the next step of his experiment, what he did was, he took S strain heat killed and R strain alive. Heat killed S strain, he took S strain, and this S strain was heat killed. And along with this, he took R strain, and this R strain was alive. So he took dead, heat-killed S strain and alive R strain. And when this combination, in which, see, the one which does not cause the disease was alive, whereas the one which causes the disease was heat-killed. So when he took this combination and when he injected this into mouse, when this was injected in the mice died. The mice died of pneumonia. When he took only S strain, which was heat killed, and injected into the mouse was alive. Because this S strain, which was harmful one, it was heat killed. So could not show any effect in the mouse, and the mice remained alive. But when the same heat killed S strain, R strain, which was alive, and then injected that, see, the alive one was R strain, which was harmless, which did not cause disease, but still the mice died. Whereas the one which used to cause disease, it was heat killed, but still the mice died. Why this happened? So what conclusion he gave on the basis of this experiment? He gave a conclusion, and that conclusion was, based on this experiment, he gave a conclusion, and the conclusion was of transforming principle. Conclusion was of transforming principle. And according to this, something from the S strain, some biomolecule or some principle or some agent from S strain, heat killed S strain, transformed the R strain into S strain. That means R strain was transformed into S strain by some transforming principle. Something from this heat killed S strain changed the R strain into S strain, and then this S strain caused the disease and the mice died. This happened in this, and this way he gave the transforming principle. That means, according to him, according to Griff, something from the heat killed S strain transformed the R strain into S strain, and this something from the heat killed S strain transformed R strain into S strain. Okay, that means in the presence of that particular principle, that particular biomolecule, that particular agent, the R strain developed a polysaccharide layer around it and got changed into this R strain. Just a minute, children. The R strain, in the presence of some transforming principle or some biomolecule or some agent from S strain, got changed into this R strain, got changed into S strain, and this caused the disease, and the mouse died. And he gave this name, this principle, as the transforming principle. But Griffith did not know that what is that particular thing from heat killed S strain which transformed R strain into S strain. What was that? So for that, another group of researchers, they did some experiment, and that group of researchers was Avery, McLeod, and McCarthy. So this same research, which was done by Griff, that was carried on by Avery, McLeod, and McCarthy. So the research of Griff, research of Frederick Griff. It was carried forward and McCarthy. It was carried forward by this Avery, McLeod, McCarthy. Their full name is Oswald Avery, Colin McLeod, and Macklin McCarthy. And what was that? It was biochemical characterization of transforming principle. They named it biochemical characterization of transforming principle. It was biochemical characterization of transforming principle. They further carried out the research of Frederick Griff by finding what is that particular substance which has transformed the R strain into S strain. So they carried forward the work of Griff. So for that, what they did, what is that? Whether, see, for that, what they did, they knew that it can be either DNA or it can be RNA or it can be protein which would have transformed the R strain into harmful S strain. Now they wanted to know out of these three, what is that? What is that? So they took out that and they started functioning. They started working further on what Griffith did. So first of all, they used RNAs. RNAs. No, first of all, they used proteases. Proteases. Proteases, what do? They digest proteins. That means when proteases are added, the proteins remain, the proteins become non-functional, they do not function. They do not, they remain, they become ineffective once proteases are added. This first they added proteases. When proteases was added, the protein became deactivated or they got deactivated. Only two things were left: DNA and RNA. Only these two are left because this got digested. Now, when this was injected in the mouse from S strain, this was taken from the heat killed S strain. Heat killed S strain. S strain. When this was taken from the heat killed S strain, because proteins were digested by adding proteases, only two were left, DNA and RNA. So when this was injected in the mice died of pneumonia. Mice died of pneumonia. Mice died of pneumonia. Now they came to know that it is not protein, because had it been protein, they came to know that something out of these two. Then what they did, next time they added RNases. RNases. So RNases, what do? They digest RNAs. So RNAs are not there. Now there was only proteins and DNA. Proteins and DNA was there. When these biomolecules, these were injected in mice, the mice died. Mice died again of pneumonia. Now they came to know that it is not even RNA, neither it's protein. Now this time they probably they came to know that it is DNA, which is the transforming principle, or which transforms the harmless R strain into harmful S strain. So this time what did they do? They added DNA. DNases, and DNases digest DNA. Digest DNA. So DNA becomes ineffective. Now when they injected this RNA, DNA, proteins, when this was, but this time the DNA was ineffective. So when this was injected, DNA was ineffective this time because DNAs were added. The mice was alive. The mice was alive. The mice was alive. Then they concluded that it is DNA which is the transforming principle. That means DNA from S strain. they migrated into the R strain and they transformed the DNA of R strain into harmful S strain. When the DNA of harmless R strain got converted into harmful S strain, a shiny polysaccharide layer was formed outside the rough strain, and they got converted into S strain, and finally they caused the disease and the mouse died. So they came to know that it is DNA which is the transforming principle, which is the agent which is responsible for converting the R strain into S strain. So this was the experiment which was performed by Avery, McLeod, and McCarthy. Now comes the next experiment, and that was the genetic material is DNA. Now they wanted to know, what is this DNA? Is it genetic material or what is it? So for that, another set of researchers, which is Hershey and Chase, they worked. Okay, so it was Hershey, Alfred Hershey and Martha Chase. Alfred Hershey and Martha Chase. They experimented to know that DNA is the genetic material in the year 1952. So they experimented in the year 1952 to find out whether DNA is the genetic material. DNA is the genetic material. Okay, so they worked, and for this they used radioactive sulfur and radioactive phosphorus. The two substances they used was radioactive which was S35 and radioactive phosphorus, which was P32. This was used for culturing the bacteriophages. They use bacteriophages. They use bacteriophages. What are these bacteriophages? These bacteriophages are the viruses which infect bacteria. These are the viruses which infect. These are the viruses which infect bacteria. So it was used. Now, what did they do to start on with their experiment? They grew some viruses on a medium that contained radioactive sulfur and some on the medium that contained radioactive phosphorus. So radioactive sulfur and radioactive phosphorus you used. That means they used what they did. Some bacteriophages were grown in S35 and some bacteriophages were grown in P32. Some were grown in S35, some were grown in radioactive phosphorus, which is P32. Now, as you know, sulfur is a part of proteins. Sulfur is a part of amino acid. So this will label the protein part of the bacteriophage, and phosphorus is a part of DNA or genetic material. So it will label the DNA or it will label the genetic material. The radioactive sulfur will label the protein code, and the radioactive phosphorus will label the DNA. Now, bacteriophages are like this. Now, I'll show you. In this case, because sulfur, see, bacteriophages, they have a protein coat like this, as we have done this in class 11. And then we have these tail fibers of these bacteriophages. And inside we have the genetic material, which is DNA in case of bacteriophages. So this is the protein coat. This is the protein coat. And this is the DNA or the genetic material. So this S35 is going to label this, and P32 is going to label this, because phosphorus is a part of DNA. We have got phosphorus in DNA, so it will label the genetic material, and protein is going to label the protein code. Now, what happened? What they did? For that, I'll erase this diagram. Here. Now, what happened? I'll draw the protein part with black color and the DNA part with green color. Now here, this was when they cultured the bacteriophages in a medium of S35. In a medium of S35, so what got labeled? This is the thing which has got labeled. The protein code, it got labeled. The protein code has got labeled. The protein code got labeled, whereas the inside one, the DNA, it remained as it was. And in another experiment, they cultured the bacteriophage P32. In that case, what happened? This is the protein code. Okay, so protein code was labeled with the S35, whereas the DNA, it got labeled with P32. So DNA got labeled with P32, whereas the protein code got, because this was cultured in the normal medium, so its DNA was not labeled. It will be normal DNA. It will be normal DNA. Okay, this was not labeled, only the protein code was labeled. So that normal, I'm going to draw with another color, the red color. So that the normal I'm going to draw with the red color. Here. So this is the normal one. Okay. Now here the normal one is red. This is the normal color. Labeling is only on the red is the normal color. Labeling is only on the DNA part, which is green in color. And here the labeling is only on the protein part, which is black in color. So here this we have found. Now the three steps which took place was infection, blending, and centrifugation. The three steps which were carried out was infection, blending, and the third was centrifugation. Okay, here the bacteriophages in which the protein code was labeled with S35 black in color. These were also allowed to infect the bacteria, and these bacteriophages in which the DNA, the genetic material is labeled, these were also allowed to infect the bacteria. So when they were allowed to infect the bacteria, what happened? This is the bacteria. This is the bacteria they have infected. This is the bacteria they have infected. Okay. This was the first step, which was infection. Now what happened? When infect, they attach to the surface. And what do they send inside? They send inside the genetic material. The genetic material is injected inside. So in them, this genetic material was injected inside, which was normal, which was not labeled with the radioisotope. And in another case, which was there, in which the genetic material was labeled, in that case also, this was allowed to infect. So the labeled genetic material was injected in. The genetic material was labeled with P32. The next step was blending. Now, when the genetic material went inside, it was blended. It was allowed to mix. And after mixing, what happened? It was centrifuged. Centrifuge means taken in the test tube and allowed to rotate at certain speed a fast speed, and in that case what happens? The heavier thing, it settles at the bottom of the test tube, and the lighter it becomes a supernatant. So what do we get? We get two things in the test tube. One is supernatant, this one, the top one, the supernatant, and the bottom one, the sediment. This is the sediment which we get, and we get this supernatant, the lighter material, supernatant. Supernatant and the sediment. The sediment had the bacterial cell with the DNA of bacteriophage. Sediment had the bacterial cell with DNA of bacteriophage. They had this. Okay. Whereas the supernatant had the protein covers, the protein coats. Supernatant had protein coats. I think, is it clear, or shall I tell you this again? The supernatant and sediment part. Is it clear from the diagram which I have made, or shall I tell you again, beta? Let me know. Shall I make another diagram to make you understand this supernatant and the sediment part? Yes, bache, is it clear, or shall I make it again? Okay, it's clear. Shagun is saying it's clear to others. Okay.